Today, I'm going to be talking about my PhD work that I was able to do at Christian Group. I was part of it at University of Chicago, and it's about investigating mechanism of calcium entry in lysosomes. Uh, Before jumping into the presentation, I was just thinking of giving a quick acknowledgement to the my PhD lab. Primarily, Amina Christian, my PhD professor, and Matt Zajac, who was the primary author, actually doing this work, and also the all the team members who actually brought in all the great expertise in kind of handling this topic and finishing it through especially during COVID times. So it was like a really great to work remotely as well. All right. So this is how I kind of broadly divided my talk today, into three sections. The first section is going to be I'm just going to introduce my point of view about what I feel about calcium and intracellular organelles. And also I'm going to touch on the prior tech that our lab, Yamuna's lab, actually had designed, which is the DNA-based calcium reporter, which I'm going to kind of show you that was key in developing other importer assays. In the previous work, which kind of led to do a very targeted RNAi screen that landed us to find the lysosomal calcium importer. And the second part, I'm also just going to talk about how actually we're using different mutants to kind of understand and solidify that the LCI, which we found is indeed the calcium importer in the lysosomes. And finally, our efforts through calcium imaging as well as electrophysiology to convince that the LCI or TME165 is indeed the calcium importer in the lysosomes. All right. Okay. So coming to the first part, to kind of like this particular audience, I don't have to talk about the importance of calcium levels. But what I find quite interesting when you look from intracellular organelles, how the calcium levels are robustly regulated. At least, for instance, at the plasma membrane level, you have 2 millimolar or 1 to 2 millimolar extracellular, which are actually kind of primarily done by PMCA1 ATPase, which effluxes the calcium out. Similarly, you have ER, where the Sarca kind of maintains 1 millimolar of calcium. Golgi, where SPCA ATPase kind of does something similar with hundreds and 200s of micromolar levels. Mitochondria has a different kind of uniporter to kind of maintain or set 10 micromolar. In Yamuna's lab, over a decade, we kind of been obsessed about mapping intercellular ions in different organelles. And we were primarily quite interested in the early endosome to lysosomal pathway. And in this way, we have systematically mapped protons, chloride. My PhD work was primarily on membrane potential. So we have like a wide array of numbers and such. So in 2018, we were kind of interested in doing calcium, intercellular calcium. And we kind of found out that, to our surprise, when you have extracellular quite high, the early endosome, late endosome is rather low. So there is a massive calcium leak once it kind of forms the vesicles. And then at lysosome, mysteriously, we have a very high amount of calcium, close to 300 micromolar. And there are a lot of interesting research that have been done who have been looking at how the calcium in the lysosome gets refilled, but none actually have kind of pointed to a specific protein that could be doing a similar job that where we saw like other proteins have been doing in other membranes. So moving on, I just also thought I'll give a quick introduction to the kind of the primary technique that Yamuna Lab uses in looking at ions. So these are DNA-based chemical reporters, which kind of primarily, basically a duplex DNA strand, and it has three modules, usually a fluorescent sensitive dye, which senses the analyte of choice. So at least today, you're going to be looking at calcium sensitive dye. And we have a normalizing dye, which is insensitive to the analyte that you want to look at. So in this way, you can do a ratio of these two fluorescent probes and kind of normalize to any sort of concentration differences. After all the different organelles are quite diverse. And finally, we have this targeting module, which kind of helps localize the sensor to different locations. At least for today's topic, we're going to be focusing more on the early to lysosomal pathway. So we use just the bare backbone of the DNA, which is negatively charged, as a ligand to interact with scavenger receptors that are usually heavily expressed in uh, macrophages and immune cells. So in this way, the plasma membrane receptor binds the DNA probes and actually kind of traffics along to the lysosome. And in this way, you could actually study the lysosomal ion. All right. So the reporter that actually we had developed in 2018 was this pH correctable DNA-based reporter for organelle calcium. So one of the biggest challenge in the field for calcium sensing, at least in the lysosome specifically, is that almost all reporters are pH sensitive as well. And because lysosomes are quite acidic, around 4.5 to 5, most of the calcium sensors kind of fall at this location where it has issues. So for instance, we specifically chose a version of rhodamine 5F, so where in such a way that the affinity for this is in the right range in the lysosomes. So one difference in this probe is that it is, instead of two dyes that I showed you before, we have three dyes, where we have a calcium sensitive dye, insensitive dye, and we also attach a pH sensitive dye directly to the probe. So in this way, if you could imagine wherever you're looking for calcium, you could actually first measure the pH and then do a correction on to understand what the calcium is. And here we have, this is a calibration profile just to show you that how the pH trace, which shows this beautiful sigmoidal 3D surface curve, is independent of the calcium levels. Whereas if you do a similar trace with calcium, you could see two things: that as the pH changes, you could see the KD of the probe changes. At the same time, the fold chain changes. So hence, it's kind of important for us to keep track of all these factors for us to do correction. And our primary choice in this work has been the model organism C. elegans. For one thing, it's transparent. It gives us more endogenous look at how cells and organelles behave. And now that I've kind of done some work with Carson Lab, where we look at quite fast dynamics, I think retrospectively, this is a very big advantage that these specific cells that we look at, called coelomocytes, these are immune cells, and they are actually static over its period of time. So they're stuck to the extracellular matrix, and it doesn't move much. So in that way, we can do really good imaging and get quite a lot of information in a more endogenous level. So here is just a typical DNA-based reporter that when you do kind of inject using a microinjector in a pseudocoelom, these cells, coelomocytes, uptake these reporters quite nicely. And you could see, if you can zoom in, you could see it's kind of localized in these enlarged vesicles, which are basically lysosomes. And Yamna's lab have been studying this for like a few years now. Okay. If so now, if you kind of do a similar experiment with a calcium reporter, you would usually get three images, but now you can play around with it. So T by A is the image where we kind of do a fold chain ratio between the pH sensitive dye and insensitive dye. So this kind of gives you a map of how the two dyes ratio looks like. Because now we have a very detailed calibration curve, we could convert that to a pH map. So now we could see, first thing is like not every endosome is in the same pH level. You have this heterogeneity, which kind of makes it more complex to do calcium in a more average way. So hence this sort of a pH correction at single endosome level seems more appropriate. So now that we know pH, we actually can also find a KD map. So this shows that how every endosome has a different KD for the calcium sensing ability. And in this way, with the help of other factors like fold change, we can actually make a calcium map and actually quantify calcium. All right, so why am I talking about 2018 work? It's because at that kind of, in that effort of using the sensor, we kind of were able to uh, solidify this triple layered lysosomal calcium importer assay, which we recently used it to actually do a targeted RNAi screen to look for calcium importer. So let me just take a little bit of time to explain what is this assay. So it's actually a three levels. We go all the way from looking at calcium in the organelle to whole organism level at the viability. So the first column is more like if you can imagine a normal lysosome with a normal size in a very happy world. But now we actually add into that mix a transgenic worm where the CUP5, which is actually a TRPML1 ortholog knockout. One thing is that the homozygous knockout is lethal. So we kind of use heterozygous knockout to do our studies. We notice, and I think it's reported that they have enlarged lysosomes. And we suspected that they would have elevated calcium level because we are kind of removing a calcium efflux channel. And if you kind of like let the heterozygous worms be on the plate, you do see a reduced population because the homozygous eventually dies. Now, our rationale was that in this background of CUP5 knockout, if you actually, it was a more primitive hypothesis that we had, but that if in case you actually remove the calcium importer, in a rudimentary way, you're kind of bringing out a homeostasis of calcium exchange. And we were kind of expecting if that works, we would see a normal size and rescue the viability or the viability of the worms. So that's what we did in 2018, and we kind of like use a sensor to check this. So first thing is that if you like do the average pH and calcium measurements, in the wild type worm lysosomes, you see the lysosome to be around 5.3, and the calcium levels are on 15 micromolar. Uh, and of course, like because the worm's its temperature is maintained at 16 degrees Celsius, and I think their mechanisms and uh, physiology is different. So the values of the calcium and the pH are at least few order different than mammalian cells. Now, back then we were kind of also looking at a specific protein called CATP6, CATP6, which is an ortholog of ATP13A2. We were kind of, because they were reported to have some sort of a cation transporter activity, we suspected if this is somehow involved in uh, facilitating calcium import. So when we did the experiment, here what we're doing is we're knocking down CatP6. We do see a slight change in pH by a unit, but we do see quite a drastic decrease in calcium. And that made us think that CatP6 might be actually a key player in calcium import. And if you go by the assay, if you look at the CUP5 knockout worm, they do have quite a high amount of calcium, close to 50 micromolar, almost 2.5 folds higher, and also a unit of 0.1 unit uh, decrease in pH. And what, as suspected, if you, on top of the CUP5 knockout, if you knock down CatP6, you seem to recover both pH and calcium level. So that was our rationale that, okay, you could do a semi homeostasis if you kind of like go after both efflux and importer at the same time. And looking at the next layer, which is the size, we kind of see a similar thing where here we use a specialized transgenic worm, which kind of the muscle cells are constantly producing GFP and in the kind of in the pseudocoelom. So these immune cells are actually eating it up. So in a way, you're constitutively labeling the lysosomes in these worms. So if you look at the size of a wild type worm, we are kind of scoring for a large lysosomal size of a specific cutoff ratio. You could see when you have a CUP5 knockout, they are actually insanely big. You could see close to around, and this is in the background of CUP5. And then on top of that, we do a double knockdown of either empty vehicle or CatP6. Previously, people have reported that multi-drug protein MRP4, multi-drug resistant transporter, could actually has shown to be reversing this CUP5 phenotype. So we also wanted to include that. Just curiosity-wise, is it really affecting the lysosome? CatP5 is a homolog of CatP6, but there are reports that say that CatP6 are present in the coelomocyte, whereas CatP5 are more present in the gut. So from the size experiment, we kind of get a similar idea where in the background of CUP5, which shows a quite enlarged lysosome, if you or kind of knock down CatP6, you approach normality in the terms of normal size. Again, it's kind of suiting the second layer where it goes by the similar principle that if you kind of remove both the efflux and the importer, you could actually, to some level, get normality. And finally, the third layer is the this is the survival assay that we do. Because I mentioned that the homozygous Cup5 is lethal. On top of that, if you actually knock down LC, any sort of an importer, uh, we kind of hope we would have a rescue of this viability. And that is what we see. I think, as like previous reports said, MRP4 kind of does a rescue. We're not exactly sure. We don't believe this is at the lysosomal level, but the CatP6 does this rescue. And if you kind of put all three factors together, it kind of like quite convincingly shows that CatP6 is involved in calcium import. But again, this was in 2018, and there are a couple of reports which have shown that ATP13A2 is a different kind. It kind of transports different analytes such as spermine and spermidine, and there are other reports also which says it transports potassium and proton. So this sort of a CatP6 or ATP13A could be a regulator of calcium import. All right. So with this solid assay of doing calcium import, we kind of like did a targeted RNAi in the worms. That's another advantage of worms is we could just feed bacteria with a specific RNA that they would just eat up the bacteria and you kind of get the knockdown. So it was a great way to kind of do this RNAi and look for just a survival assay. So we kind of look at you're scoring the brood size difference between RNAi and control. And the biggest hit that we had, which was Y54F10AL1. If you can just do a quick search, it is nothing but an ortholog of TMEM165 in humans and GDT1 in yeast. And these were actually previously reported to show calcium importability, but in Golgi, and they primarily studied it in oncology. And so we kind of, at least from the assay perspective, we found it quite interesting that we landed on something which was actually a calcium importer. Now, the biggest question we had next was, is it on the lysosomal membrane? Uh, all right. So now that we know I think we kind of like not minimize this name to LCI1. So just moving forward in worms, we just call it LCI1. So if you kind of do the lysosomal size assay, uh, you could see like in the background, we have like a 40% larger lysosomes when you have a Cup5. So this is not a knockout. It is a missense mutation, which kind of results in inhibition of the function. Uh, so it also shows enlarged endosomes, lysosomes, which we can actually see. And then on top of that, we did CatP6 as a control from our previous work. It seems to rescue the size. And when we do the LCI1 knockdown, we see again a similar rescue, suggesting that similar to CatP6, LCI1 could be involved in facilitating the calcium import. So that was again in the transgenic worm. We went back to the wild type worm. Now we inject a marker for lysosomes such as dextrin, which labels the lysosomes over time. And now actually we have a knockout worm, LCI1, and we kind of look at the size. So now again, the rationale, or at least the understanding was uh, before the Cup5, which was an efflux channel, and kind of inhibiting that or knocking that down increased in size. Now you could expect a similar sort of a reaction when you inhibit or affect import channel or transporter. So that is what we see when you have only the LCI heterozygous knockout. You kind of observe enlarged endosomes. And if you do a Cup5 knockdown on top of that, you actually, again, approach normality. Now, another interesting thing is if the similar to Cup5 homozygous mutant knockout, LCI1 homozygous is also lethal. So there seems to be a quite of a balance between the import and the export. All right. So with that, we kind of got an idea that, okay, LCA1 is of interest. And thankfully, there were previous literature work, which was also actually pointing at few different mutants. So in the next part, I'm going to be talking about how we explore those few mutants, at least in the case of localization or in perturbating activity. We kind of like try to elucidate what is happening at the lysosomes. All right. The first question we had is like, okay, the problem with working with something which is in the Golgi or ER is that you could definitely be catching some sort of a secondary effect at the lysosome, and that could be a big misstep. So the first thing we want to like really be sure is there LCI in the lysosomes? Although like there were reports before which shown that certain mutants prefer to be lysosomal. So we knew that they are designed to go to lysosome, but is it actually functionally sitting there? So the first thing we did was just like to look at the colocalization of like a lysosomal marker such as FTC-dextrin that we pulse chase to lysosome. And so these are the green spots are lysosomes. And over here, we kind of overexpress LCI fused with DS red. And you can clearly see there is a huge Golgi population, but also there are these punctase which colocalizes with FTC-dextrin, suggesting that you have a fraction of LCS at the lysosomal membrane. Now, those are the overexpressed scenario. And here we kind of like are looking at endogenous level. And we also just wanted to kind of expand the lysosome to introduce a little bit of a resolution to see if they are on the limiting membrane. And so we kind of treat the HeLa cells for a few hours with Bacillin-1, which enlarges into lysosomes. In a way, it kind of fuses endosome and lysosome. So it's not very accurate in terms of what organelle it is, but it's definitely endolysosome. And you kind of like stain with an antibody for LCA. So you're looking at the endogenous level. And then we also stain with LAMP1, which is known to be present in endolysosomes. So this kind of gave us some confidence that, okay, there is a fraction that is present in the lysosome. And now there are some, again, like previously people have investigated mislocalizing variants of this protein, TMA165. The two cases that we really liked was the G304R, which is kind of considered to be a Golgi favoring mutant because if you express this, it prefers to stay in the Golgi. And then you also have R126C or 126H, which is, so this YNR NRL motive is considered to be a lysosome localization motive, but mutant of or a mutation of R to C or H actually makes it more lysosomal favoring. So in this way, we now kind of have two mutant which they can switch specifically to one organelle than the other. So we went back to our three-layer assay and we trying to understand if can we understand something about were we able to selectively perturb the lysosomal calcium. So again, you're looking at N2, which is the wild type worm, basically the normal worm. You're looking at like we're going to normalize to the total number of worms. So this is the survival assay. So if you knock out LCI1, of course, heterozygously, you see a decrease in the population brood size. But now we are kind of overexpressing or expressing extrachromosomally wild type human LCA. So it's in a way like we're trying to see if the human LCA can rescue lysosomal or this whole survival assay in C. elegans. So when you express that, it kind of approaches back to basal level, suggesting that yes, LCA from human can actually perform a similar same function as the C. elegans version. Now, if you do R126C, which is the lysosomal favoring mutant, it approaches closer to the basal level that it rescues in a significant way. And if you do the Golgi mutant, which is Golgi favoring, so now all the LCI in this case is the Golgi, you don't seem to actually rescue the viability of the worms, suggesting that it kind of you need the functional aspect to be at the lysosome, or this death that we are seeing is due to some sort of a lysosomal disorder situation. Similarly, if you do the size assay, we get a very similar outcome that G304 is actually the size is still enlarged, it's not reducing, whereas wild type LCI or the lysosomal favoring one actually becomes very close to the wild type, suggesting again a similar thing at the lysosomal level. And finally, too, if you kind of do the calcium mapping in these worms, we don't see any difference in the pH. More or less in all the cases, it seems the pH is around 5.3 plus or minus 0.1. But in the case of lysosome, of course, you could see a massive decrease with LCI1 knockout. And when you kind of like reintroduce wild type or the lysosomal favoring one, you rescue it completely. And surprisingly, even G304R seem to outperform compared to previous assay. So this is something where we actually right now we're not completely sure. We suspect there is some aspect of Because these are heterozygous, there could be some sort of like a dimerization between two mutants that could result in something similar. At the same time, or we could think that the calcium import or the calcium level is more sensitive compared to the other assays. All right. Uh, so with this, actually, we kind of got like a little bit more confidence that it is not anything to do with affecting the Golgi that is showing this sort of a rescue or calcium changes. It is indeed something directly that we are operating at the lysosomes that is actually changing all this effect. Uh, and we also kind of want to directly touch on it. So we wanted to go after selective locations that we could mutate. But we were also very cautious. We didn't want to spend too much effort in it. So we kind of like compared and the TMA165 across species as well as with VCX1, which is like a very well-known calcium proton exchanger in yeast. How do the structurally and sequence-wise they compare? Uh, so one interesting thing is, of course, I think in the field also people have looked at the TM65. They have this acidic loop on the cytosolic side. With at least in the case of VCX1, they have actually shown that it is pivotal in binding calcium on the cytosolic side for it to transport. So we kind of wanted to touch on that. So we did one mutation point E188A at that location. And also, one of the key aspects of TM165 is this motif that is present in transmembrane domain 4 and 5, which, again, if you do a structural comparison modeling with VCX1, it kind of comes very close to uh, the actual calcium binding domain in VCX1. So again, but simple logic that glutamate would potentially bind the calcium, we again mutated the glutamate 108A and 248A to A. And another important thing is that people also have reported that this particular mutation, missense mutation, actually in humans have caused congenital disease, a disorder of glycosylation, suggesting that this is definitely affecting the protein. So these three were of our interest, and we kind of like made these worms and moved on with our assay. So again, looking at the brood size, you could kind of like see compared to the wild type and the knockout, E108A and 248A is actually not able to rescue it, and E188 is partially rescues it, does a good job. It seems insignificant compared to N2. So to at least us, we seem to have two interesting players to look for. And if you look at the size, we see a similar scenario where E108A and E248A is actually not able to rescue, suggesting that they lost the function, whereas E188 actually is able to decrease the size, suggesting that the acidic loop might not be that important for it to for this particular function. And finally, if you look at the calcium levels, this was again a surprise. Although we have seen before that calcium level is quite sensitive, the assay, we kind of didn't see a similar trend. We did notice all three actually rescuing the calcium levels. And this is where kind of we will look a little closer, and we did have a complication, which is that again, this is part of the sensor. The sensor has a sensitive range, and it is at a specific pH, it is not able to sense below 100 nanomolar. So if in case we have any sort of a value that is close to 100 nanomolar or below, it's going to actually have quite a lot of error, and we technically cannot be pulling it in. So if you can just look at, so we made like a uh, histogram where we kind of like do comparison between N2 and LCI knockout. You could kind of clearly see that a huge population, 50% of them are below the detection limit of or the sensitive limit of the sensor. And so although like we shouldn't be involving this in our uh, uh, average calculation, but I think it kind of clearly shows you that the population shifts so much that it actually goes below our sensor range. So ideally, we should have like better sensor, but I think this kind of still bruises the point that not having LCI actually really drops the calcium levels in the lysosome. But now, keeping this information, if you go back to the three different mutants, you would see that E248A seems to be actually, and E18 both actually shows quite a low levels of calcium in a huge population of endosomes. There are still few endosomes which actually have the right amount of calcium, but by looking at the levels, which is much lower than zero point 0.1, you could actually land on E248A as a favorable choice for us to kind of like study more in terms of how is it affecting the calcium transport. All right. So with that, we kind of like look at how calcium entry is affected or taken facilitated. So here we actually have TMEM. So we took HeLa cells. I've been showing you calcium levels on the C elegans, but now if you look at the cells, these are HeLa cells. We're looking at the wild type versus LCI knockout, and you could easily pick up that there is a difference in the pH. In the case of knockout, it is more acidified, and it kind of goes with the calcium proton transport theory or hypothesis that, okay, you're removing a calcium importer is actually acidifying the average population. And looking at the calcium, at least again, the average, we didn't pick up a difference. But then that is also because we have the same sensitive issue that in the case of a knockout, most of their lysosomes are empty and it's going below the sensitive limit. And therefore, it's actually higher than wild type. So that was the steady state. We could see a difference at the calcium. We could see similar acidification in the pH. And we kind of wanted to try if we could capture in a more dynamic sense. So for that, we kind of use this selective way of introducing cytosolic calcium fluctuations. So we add like extracellular 100 micromolar of ATP, which with the help of PLC produces IP3 in the cytosol, which induces the calcium efflux from the ER. And we kind of just did a check to see if does all cell respond at the same time and so on. And this kind of map does that around 1 minute time point is a great point to look at right after adding ATP. Now, one of the drawback with having three dyes on the same system is that they all have different bleaching points. So it is quite difficult to do very dynamic mapping. So we resorted to just doing two time points. One is pre and another one is after adding 1 minute of ATP. So now in the left, what you're looking at is basically the cells which are treated with the calcium sensor. So we are looking at endolysosomal pH and calcium. And what we are comparing is before and after adding ATP. And we only have selected those endosomes that we can kind of reliably tell which is before and after. So in a way, you kind of like these points that we are connected is kind of the same lysosome during the 1 minute time window. So this is wild type cell. So one surprising thing we noticed was that in the case of pre and post, most of the points were actually becoming acidic, which was not the case in the before, because before it was a knockout scenario where, so again, if you're going by a calcium exchange theory, we were kind of expecting when you actually have high calcium in the cytosol, we would actually see neutralization or basification of the pH, but it seemed to be going a little other direction. But the calcium did increase. So most of the endosomes are actually increasing its calcium content. Now, when you do the same thing in a LCI knockout cells, you kind of like don't see any significant change, which kind of goes with it. But we kind of try to understand what this means, because if you do a dynamic change, why does the pH acidify when also calcium increases? So one other advantage of having the sensor is that you could actually map both calcium and pH at single endosome level. So in a way, you could make like a 2-ion map so that it instead of looking at average pH and average calcium, you can get more information about how things are changing. So over here, we have selectively drawn orange for something where you can see an increase in calcium while there is a decrease in pH, which is against the transfer exchange theory. And the blue is something where you can actually have increase in calcium with increase in pH, which is what you would assume if there is like a simple exchange. So this is where there are I think this is where there are complications when you look at systems with a more native state. For simple thing is that there could be an increase in pH or decrease in pH just because there is increase in calcium could be something similar to how when you add calcium to EGTA, the pH is becoming acidic. So you have like a different buffering change in the lysosome, which could just increase the or decrease the pH. And that is what we are picking. Because in the case of LCI knockout, we actually don't see something like that. So there is a good chance that this sort of like a calcium buffering change, changing the pH, could explain why we have this sort of a nature. But then this also kind of introduces this point that every time there's a calcium increase in the lysosome, you could potentially also have an acidity change, becoming more acidic. And does that mean having an acidic lysosome improves calcium input? So we had all these questions with this sort of a result, and we didn't want to just directly take out that, okay, this is all exchanging protons and calcium. So keeping those in also like, so we want to double check if there is an effect of pH in the calcium changes. So we kind of resorted to a more standard assays the field does, which is looking at the cytosolic calcium. And we kind of like in a way trying to look at does lysosome when there is a calcium surge in the cytosol, how good does a lysosome mop up the cytosolic calcium? So here, mark is basically the LCI knockout. So you don't see any LCI over here. Uh, so in the untreated case, you kind of see at Tmax, which is around like 1 minute, the maximum surge, and then it kind of like plateaus down at 2 minutes. When you add baflomycin, you don't see any change in the knockout as expected. In the case of wild type, you could see that it surged and then it kind of like reaches close to pre-treatment around 2 minute window, whereas in the presence of baflomycin pre-treatment, you actually affected that sort of a mopping of cytosolic calcium across lysosomes. And we also actually did R121C. So again, the expectation is that now you're putting the LCI more exclusively in the lysosome. Does that do a better job in mopping up? And if you want to like look at the quantification, you could kind of like start seeing that you don't see any difference in the uh, knockout, which is the mark. There's no effect on the baflomycin. But in the case of wild type and the lysosomal favoring mutant, you could actually see that adding baflomycin is perturbing the mopping up of calcium from the cytosol. So with this, we kind of did have an idea that, okay, there is a pH effect, which people have also reported that in order for calcium to refill into the lysosome, you need to have like a pH gradient. At the same time, we are kind of looking at, is this a exchanger or a transporter scenario? So for that, we went to electrophysiology, and the first thing we did was to see if do we have enough LCACs, or sorry, that's a type of LCI on the plasma membrane. So here we kind of just overexpressing LCI on the cells, and we do an immunostaining of EGFP because here the EGFP is on the cytosolic side. Oh, sorry, on the luminal side. So we're kind of looking for EGFP at the plasma membrane to just confirm if we have signal suggesting that there is enough proteins for us to do the clamping. That is what we see. We see in the case of overexpression, we can actually start seeing this anti-EGFP signal, which we don't see in the basal level. So the first thing we did was, okay, we kind of wanted to play safe and we didn't want to have any sort of ions that could potentially disrupt our uh, signals. So we kind of exclusively use NMDG as the positive, and then we also change the calcium exclusively calcium hydroxide. So in this way, if we kind of like specifically look at the traces, having more calcium in the cytosol or in the pipette solution, we see a more higher outward current. At least over here, we're kind of plotting at plus 100 milliwatt. And we see that higher the calcium concentration, higher the current, uh, outward current. Now, the question that is this pH dependent? We changed the pH on the cytosolics on the extracellular side, which is the bath solution. And over here, we kind of like try to make the lysosomal gradient somewhat similar or maybe closer to similar to the lysosomes. So instead of actually adding more in the uh, cytosol, which is what you should have done, we kind of like actually try to recapitulate lysosomal feeding scenario. So hence, we added like 100 times more on the extracellular side. And we were kind of like surprised to see still like a very good outward current, especially at a very acidic pH at 5.5. We could see pretty good current, and it kind of drops. It goes very low at 7.5. And we also introduced the E248A, which we kind of reason to be a really good loss of function mutant. At acidic pH, it kind of like decreases and goes to the basal around 7.5 level, suggesting that the pH does have an effect in the calcium transport across the uh, transporter. We also, okay, these are the plasma membrane, and we also wanted to really check if something similar can work in the lysosomes. So in the case of COS7 cells, which actually don't have endogenous LCI, we overexpressed LCI, and we expanded the endosome using vacuolin, which is what in the community actually do to increase endolysosomal size, and then they selectively uh, uh, rupture the cell and actually patch the lysosomal membrane. So first thing to notice is that we do see the LCI, EGFP on the limiting membrane. And of course, we do look for something which is overexpressed, and we patch a lysosome where you can actually see the LCI on the limiting membrane. And in this way, over here, we kind of resorted to actually having a very uh, endogenous level of ions, unlike the plasma membrane, to at least compare between a Golgi pH and a lysosomal pH. So we kind of compared what sort of a current differences do we see when we actually clamp at pH 6.2 in the lysosome versus pH 4.5 in these organelles. And you can see like at pH 4.5, the current levels are higher compared to Golgi level pH. All right, so with that, I'll summarize uh, the talk. So the phenotypic screen, the three-layer screen that actually our sensor use kind of were able to establish, identified LCI1 in C. elegans as a facilitator of lysosomal calcium. Again, by previous literature as well as our experiments, a substantial fraction of LCI are localized to Golgi, but the transporter is, from our experiments, do show that they are more active in lysosomal membrane, and this is possibly due to higher pH gradient that lysosomal harbors. Uh, Now, the calcium store refilling has been people have done a lot of experiments like Hao-Shin Chu's lab, Schwanz's lab. They've been doing it over decades, and they have actually established quite a few hypotheses, such as Hao-Shin Chu lab actually has put forth this that it's not the pH gradient, but the ER interaction with the lysosome, or the ER needs to release calcium for the lysosome to actually fill in. So I think something having like an acidic loop on the cytosolic side, similar to VCX1, makes a lot of sense that it needs to bind calcium for it to actually import. So it seems like a very good, it goes with Hao-Shin-Chu's hypothesis. Similarly, we do see a very strong pH dependence in the calcium import, uh, going with other people's experiments where I think the calcium import is a very pH dependent phenomenon. And since the human LCI can actually restore lysosomal calcium channel function, which is what our assays have been showing, it actually might offer new ways to investigate existing disorders which are caused by defective lysosomal channels. Uh, with that, I'd like to again just thank all the past members and current members of Yemen Christian Group, the funding agencies, and thank you all for listening.
Thank you, Anand. Fantastic. Okay, I think we've got a couple of questions in the Q&A box, or a number of questions.
Okay.
Let's try and work through a few of these. A few questions from an anonymous attendee who says, "Thank you very much for the nice talk." And then a number of questions which I think are interesting. So first of all, something I was wondering about: what are the holding and reversal potentials of various organelle membranes? So what's the membrane potential across the lysosome, for example? Maybe that's useful for people.
Right. So I think over here, when they say, so at least when the native lysosomes that I have measured with, like, say, the voltage sensors that we have developed, they are actually usually in the range of minus 100, if you believe cytosol to be negative. Like, if you take cytosol to be negative, they are on minus 100 milliwatt. Uh, so
And
that's what.
a question that I had with your siRNA screens, there's
Mhm.
always the danger that you change the transmembrane voltage of the lysosome and then you get effects due to changes in driving force on ions rather than the actual calcium transport pathway. Is that a consideration with the siRNA screens?
Yes, you're right. And so that is one of actually, it could be, you can take it as a drawback, or you can actually also take it as a blessing, because if you're thinking of looking at other cofactors and other way factors that could be affecting, say, the calcium change, uh, I think having, say, some protein which is changing membrane potential or changing proton levels could also actually come in our assay. So that is why we kind of like really look for the most massive change. And then, of course, I think seeing previous literature where they have kind of put forth this Stemium 165 as a calcium uh, transporter kind of made us go very forcefully on this particular
Right.
protein.
Are endosomic pH levels continuously changing? Can you see temporal kinetics?
Like I said, because here we are using multiple dyes, and most of our experiments are actually wide field or confocal. So we do risk bleaching these sensors, and the biggest concern with endolysosomes is that they do move a lot in the 3D. So you cannot like do one snap, like just one plane, like you're doing a calcium levels, and so hence most of the time we just do one or two stars time points with all the 30 to 60 Z stacks. So hence, I think this particular question, I think we don't have the answer at the moment.
I'll drop down to a question from Veronica Loyo Chelis or Celis. LCI has a voltage sensor motif. Does this permeate any other cation apart from calcium?
Uh,
Oh,
so
that's a question. Does LCI have a voltage sensor motif? I think.
at least it doesn't have the usual voltage sensitive domains that the channels have. Uh, but to in our experiments, like at least like we were not expecting to see any sort of a current, because again we were thinking, okay, this is going to be similar to VCX1, which doesn't, it kind of like just transports one calcium and two protons. We wouldn't have any sort of like a even a current or such thing. But we were surprised to see there was like a calcium current. Again, this raises the question that maybe it has a different level of uh, stoichiometry than the VCX one. But yeah, we didn't come across any sort of a voltage sensitive domain in this. And coming to LCF, it permeates other cation. In our experiments, we maybe this is something to do with just doing calcium clamping, but we had to like very selectively had to remove potassium and sodium, because I think we did have a lot of leak currents through these ions whenever we have them.
Yeah.
So
But actually, with your patch clamp recordings, is it possible some of the current upon changing pH is proton current? It seems consistent with the driving force, right?
yeah, I think so. One of the reason we didn't actually kind of declare that it is really, we do see a proton transport, is that we couldn't actually get a very reliable reversal potential change for this thing that we're setting. It seems a little complicated. We are getting kind of a reliable direction, the way the calcium is moving, but with the case of proton, it seems a little bit more complicated, that we're not actually able to confirm with the clamping, at least, that it is indeed transporting proton. And I think with our kind of like calcium sensors and other experiments, we kind of at this moment, at least, we are kind of considering this as like a pH dependent calcium transport. Um, and also that because these are, it seems somehow like the total number of domains are only like half of what BCX is, we kind of believe there is something more complex than just a simple exchanger. So I think currently Yamuna's group is actually working on this to understand more in depth how actually LCI could be working.
Great. We've got a question from Damien Bell, and then we can return to the additional questions at the top. So Damien says, "Nice talk, Dr. Saminathan. The lysosomal patch clamp studies near the end, were these LCI transporter currents or lysosomal calcium channel currents? Or put another way, is the LCI transporter fast enough to measure calcium transport currents by patch clamp?
Um,
So were these currents carried by LCI itself, do you think?
yeah, because we didn't kind of record a similar current when, so because in Mozart we kind of did experiments in also like knockout cells, and in the knockout cells we kind of overexpress the LCI. So we kind of try to remove any non-LCI mediated leak currents or any such things. So we are pretty confident that the currents that we're looking at are the LCI. The levels are, uh, if you look at it, it's pico amp. It is quite low, but it is from a transporter perspective, it still seems a little higher. So, but the fact that I think in many cases we kind of saw currents going in the opposite direction to uh, the calcium levels. So that's where you're going to make us think that it is somehow uh, could be a pH activation that is involved in the calcium uh, transport.
Okay, let's try and make our way through some of the other questions. I know you have to leave in a couple of minutes. Maybe we can copy these and find a way
Mhm.
to. If the attendee would like to submit an email address, maybe we could get back to you on some of these. Is there a risk of introducing bias to native genetic program when using DNA-based probes?
Is there a risk of introducing bias in native genetic program while using? So these DNA-based probes are, it's pretty small. Like it usually are like 30 to 40 nucleotide. Uh, and we have kind of like selectively chosen the sequence in a way. First thing is like they have very minimal immune response, like it doesn't interact with TLR much. Of course there could be, but again we have chosen the sequence in a way where we see minimal effect. So at worst case you could have such sort of like signaling situations, but not anything in terms of genetic reprogramming and such. That wouldn't be the case.
Okay, maybe we got time for a couple more. So your pH recovery data, CAT-P6, is it some standard deviation or SEM?
Those are actually SEM between experiments.
And then in the lysosome assay, it seems there's an antagonistic trend between CAT-P5 and CAT-P6. Can you confirm that? Is that correct?
That is correct. So with the cat piece, so we strongly believe the cat p6 is present in those telomercyte lysosomes, whereas cat p5 are present in some other cell type. So hence we are seeing cat p5 does a rescue on the survival of this worm. But when it comes to the size and the calcium levels, cat p5 actually does not show a change, again suggesting that CatP6 is the one that is present in the lysosomes of the ceremocytes and not CatP5. So there could be like a tissue dependence in these homologs.
Yeah, that's an interesting idea. So you think there might be different protein expression in different tissues in that affect the lysosome differentially in different tissues?
Yeah, because it's interesting in coelomocytes. Usually, most of the homologs of humans like have these homologs and C. elegans actually have multiple copies. So at least like ADP, the ADP13A2 in C. elegans actually are like three, I think CatP6, CatP5, and CatP7, I believe. Uh, so I think
Okay.
SE is known to show this like differences in the tissues.
And with that, I would like to thank Anand for a fantastic presentation and thank you everyone for attending.